Hi guys, welcome back to my channel S Chemistry. My name is Sonal and today I'm going to talk about a very important biomolecule in all the living systems known as the hemoglobin. Now I will be doing 3 different videos on hemoglobin because I'm going to cover all the aspects of hemoglobin with respect to bioinorganic chemistry or with respect to chemistry. So students doing their bachelor's or master's in general chemistry or specifically bioinorganic chemistry, or even if you guys are preparing for any of the competitive exams, then this video will definitely help you. So let's get started. So before we actually talk about what exactly hemoglobin is, there are a few terms that we must know, or there are a few things that we must be aware of. First such term is metalloporphyrins. So I have put a structure of metalloporphyrin over here. Let's just go through the slide quickly. Metalloporphyrins are one of the most important ways in which metal ions are involved in biochemistry is in complexes with the type of macrocyclic ligand which is known as porphyrin. So we are aware that the living system incorporates a huge number of metal ions, like there is cobalt, there is zinc, there is iron, and some of these, like there is magnesium and chlorophyll. So some of these metal ions are involved in biochemistry in complexes with a type of macrocyclic ligand which is known as porphyrin, the one which I have drawn over here. Porphyrins are derivatives of porphine and they usually differ in the arrangement of substituents around the periphery. So if you have a look at the image or the structure that I have put, you will see that porphyrins are basically tetrapyrrole systems, and pyrroles are nothing but these 5-membered heterocyclic rings wherein one of the atom is nitrogen. So it's a tetrapyrrole system. Pyrroles are 5-membered heterocyclic rings with nitrogen as one of the atoms. That is your porphine or porphyrin system. And another important thing to note is when you're putting down the structure of porphyrin, you will notice that there are 11 bonds in conjugation, or you can say that the system contains 11 conjugated double bonds. Moving ahead to the heme proteins. So these are actually the small things which when you put them together you get your entire structure of hemoglobin. That's why I thought I will start with this as an introduction and then we will move to exactly hemoglobin. So talking about heme proteins, iron is certainly the most widespread of the transition metals in living systems. Its compounds participate in a variety of activities. The two main functions of iron containing materials are transport of oxygen and mediation in electron transfer chains. Okay, so iron is a very important transition metals in our system and it has major 2 functions. First is obviously transport of oxygen, or carrying oxygen throughout the living system, and second of it is mediation in electron transfer chains. So much iron is required for these purposes that there is also a chemical system to store and transport iron. The chief heme proteins which are present, or the chief heme proteins to be found, are hemoglobin. That's what we're going to talk about today. Then we also have myoglobin, and the third one are cytochromes, including a special type which is P450, and then there are enzymes like catalase and peroxidase. So this video is focused on hemoglobin, so I will talk about it in a bit, but in general if I just want to talk about myoglobin, then myoglobin contains a heme prosthetic group that can reversibly bind to oxygen, and the body uses it as an oxygen storage protein in the muscles. It is able to bind and release oxygen depending on the oxygen concentration in the cells. So myoglobin is like a storage center for oxygen in the muscles. Next, talking about cytochromes, then cytochromes are redox active proteins containing a heme with a central iron atom which is present at its core as a cofactor, and cytochromes are involved in electron transport chains and also in redox catalysis. So that is like the general function cytochromes usually play in the living system. Specifically talking about cytochrome P450, then these are like a super family of enzymes containing heme as a cofactor that generally functions as monooxygenases. Now in mammals, these proteins oxidize compounds like steroids and fatty acids, and they are important for the clearance of various compounds as well as for the hormone synthesis and breakdown in living systems. Lastly, to conclude on the enzymes such as catalase, and catalase is a common enzyme which is found in nearly all of the living organisms which are exposed to oxygen, and they catalyze the decomposition of hydrogen peroxide to water and oxygen. So they actually decompose H2O2, hydrogen peroxide, to water and oxygen. It is a very important enzyme in protecting the cell from oxidative damage by reactive oxygen species. So that is the role of catalase. And lastly, peroxidases are a group of enzymes that catalyze the oxidation of a substrate by hydrogen peroxide, or you can say any organic peroxide. Peroxidase catalyze oxidation reduction reaction by mechanism of free radical. So just to summarize, I have mentioned a few points with respect to the chief heme proteins which are seen in living systems. So finally, starting with hemoglobin, which is an oxygen transport protein. Hemoglobin is a protein which is found in the red blood cells. It gives blood its red color, and its job is to carry oxygen in our body. So first, two important points. Where do you find hemoglobin? Whenever we are studying any biomolecule, the first thing to understand is where do you find them. So hemoglobin is a protein which is found in RBCs, or in the red blood cells. The next crucial point is to know its function. Like whatever biomolecule you are studying, you have to know its location and you have to know its function. So hemoglobin, found in the red blood cells, it gives blood its distinct red color, and its main function is to carry oxygen in the living system or in our bodies. Hemoglobin has a quaternary structure characteristic of many multi-subunit globular proteins. Quaternary structure is with respect to the structure of a protein. We are aware that proteins have primary structure, secondary structure, tertiary structure, and then comes the quaternary structure. So hemoglobin shows a quaternary structure, and most important point to note over here is that hemoglobin molecule is a tetramer. Tetramer meaning 4 iron atoms are present per molecule of hemoglobin. Tetra meaning 4, and the count over here, that is 4, is associated with the number of iron atoms present per molecule of hemoglobin. Now coming to very important aspect of hemoglobin in terms of chemistry, we are going to look at its structure. The structure of hemoglobin is an octahedral structure, or you can say that it is an octahedral complex of iron in the +2 oxidation state. So we know that if it's an octahedral system, then it needs to have 6 coordination number, right? Or we can call it as a 6 coordinate system. So let's talk about that in detail. The center is occupied by Fe in the +2 oxidation state. So the central metal atom in this case is iron in +2 oxidation state. Now coming to the 6 coordinate system. The 4 corners of the square base contain 4 nitrogen atoms of the heme group. So that's why I had started with porphyrin in the beginning, because you can see that there is a porphyrin system around your central metal atom. So the base, the square of the base, is formed by forming 4 bonds to the nitrogen atoms of the heme group. So 4 out of 6 bonds are clearly visible. The 5th position is bound to a nitrogen atom of the imidazole group in histidine residue. So this is the next thing to note. Okay, I'll just highlight it. So we have looked at 3 things. Okay, first of all, it's an octahedral system. Okay, it's a 6 coordinate system. Second, the central metal atom is iron in the +2 oxidation state. And third, the central square is formed by 4 nitrogen atoms of the heme group. Okay, coming to the fifth position. The fifth position is bound again to a nitrogen atom, but this nitrogen atom is of an imidazole group in the histidine residue. That's like a protein chain. Okay, and the sixth position is empty. So that is very logical why you need an empty position, because we have just seen that hemoglobin functions or acts as an oxygen carrier. So you require an empty site wherein oxygen can come and form a bond, and that is done to the sixth position. The sixth position of hemoglobin structure is used for transporting oxygen from lungs to body cells for human beings or animals. Okay, now even though I have mentioned that in the unbound state, that is in the deoxy form, the central metal atom is in the +2 oxidation state, but there can be a debate, why not +3? Okay, so if you tend to think about it on your own without having any expertise, you might have the tendency to lean towards two options. The central ion can either be in +2 state, or maybe it can be in the +3 state. Okay, it's like +2 can get oxidized to Fe3+ and then again get reduced to Fe2+. Okay, so how do we tackle this point? How do we know that in the deoxy form iron is present in the +2 oxidation state, and in the oxy form it goes to a +3 oxidation state? So I'm going to address that particular query over here. And these figures are a very good representation of the deoxy form and the oxy form. You can see that in the deoxy form there is iron in the +2 state, and it is bound centrally 4 nitrogen atoms, and then there is a nitrogen from the imidazole group. Okay, and once it binds to oxygen, it goes in Fe3+ form or Fe3+ oxidation state when it is present in the bound form. Okay, so let's address that particular question from the second point. Oxygen temporarily and reversibly oxidizes Fe2+ to Fe3+, while oxygen temporarily turns into the superoxide ion, that is O2-. Thus, we know that iron has to be present in a +2 oxidation state in order to bind to oxygen. So basically what happens is, you cannot have initially iron in the +3 state, because it has to bind to oxygen in such a way that oxygen can oxidize iron from +2 to +3. That's why you cannot really have it initially only in the +3 oxidation state. So you have oxygen, the process is temporary and reversible. It's not a permanent, irreversible chemical bonding taking place. It's a temporary process, reversible process. So initially oxygen will oxidize Fe+2 to Fe3+, and in this process oxygen gets converted into superoxide ion. Now in superoxide ion, the form or the anion is represented as O2-, wherein each oxygen atom carries a charge of -12. O2, the overall oxidation state of it is -1. That tells us that each oxygen atom carries a charge of -12. Thus, we can conclude that in the deoxy form or in the unbound form, iron is present in the +2 oxidation state, and then only it is able to bind to oxygen, get oxidized to Fe3+, and in the same process, O2 gets converted into superoxide ion. Hence, in terms of chemistry, we can conclude that in deoxyhemoglobin, iron is in the paramagnetic state, which is a +2 system. Fe in the +2 will have a configuration of D6, and it is present in the high spin state. So if you try to plot the crystal field splitting diagram of a D6 system in the high spin, you will get this particular configuration, that is T2G4 Eg2. There will be 4 electrons in T2G or at the T2G level, and there will be 2 electrons at the Eg level. D6 high spin system will look somewhat like this. So I'm just trying to draw it so you guys get a rough picture that in the T2G level you will have 4 electrons. So this is your T2G level, and at the EG level you will have your 2 electrons. Okay, so you can see that it is a paramagnetic species with 4 unpaired electrons in the deoxy form. Coming to the oxy form of hemoglobin, now it is seen that the oxyhemoglobin is a diamagnetic system. That means it's a paired system, or all the electrons are present in the paired state. Now, oxyhemoglobin has an electronic configuration of D5. It is Fe3 plus. That means it is a D5 system having odd number of D electrons. So how can you explain the diamagnetic behavior of such a species, wherein we know that if it is Fe3 plus, it's a D5 system, it will have an odd number of D electrons. Why don't you guys just pause the video and try to think about it, and then I will discuss it in some time. Come on, pause it right away. I hope you guys did get the answer. If you all did get the answer, then please put that answer down in the comment section. And now I will discuss it with you all. First of all, we need to understand that ion in the +3 state having a T5 electronic configuration is going to be a low spin system. It is a low spin system. That means all the 5 electrons will be occupied at the T2G level. So the configuration over here will be T2G5 EG0. Now we can see that there is an unpaired electron over here, and this unpaired electron should have given a paramagnetic behavior, but it does not. Instead, the system is said to be diamagnetic in nature. So let's come to the answer. It's a very simple answer. The phenomenon is termed as anti-ferromagnetic coupling. Okay, the diamagnetism arises from the single unpaired electron on superoxide anion. We have just seen it is O2 minus, each oxygen carrying an oxidation state or a charge of -12. So the diamagnetism arises from the single unpaired electron on superoxide aligning antiferromagnetically with the single unpaired electron on Fe3 plus, that is in a low spin D5 state. So both the electrons in the unpaired state are going to antiferromagnetically couple with each other, giving rise to a paired system, or giving rise to diamagnetism in your oxyhemoglobin molecule. So that's it for today. Very soon I will upload another video on hemoglobin, wherein I will focus on the structure of the complex. As of now, today I have discussed the chemistry of hemoglobin. We incorporated a little bit of bioinorganic and coordination chemistry in today's video. I hope this video was helpful. In case you did like it, then please give this video a thumbs up and share the video with your family and friends. Thank you so much for watching. I will see you guys soon. Bye bye. Take care.